topics. And what you're going to see, so I will give a quick recap about the fundamentals in biology, about the fundamentals in molecular biology, so that you better understand why AI is so important in this field, what AI does to this field. Then we will go into the AI revolution in protein structure. So this is probably the biggest, let's say the biggest achievement of AI in science. Let's say it like this. Yeah. So besides ChatGPT and all these things that are more to the aimed at the consumer side, here we're on the scientific side, and I guess that here basically this was really one of the biggest breakthrough of AI, where AI really made science better. Yeah. We will talk about the problems of structure prediction, because of course AlphaFold is not perfect, and what else there is to do in science behind this. And then we will also talk not only about protein structure, but also the sequence level. So we will very quickly see what the sequence level is, and that you can also use AI in gene sequence prediction, where it's very like, it works rather like, let's say it like a typical LLM, but where it also basically helps scientists go further. Yeah. So these are basically the things that I will show you. So first we will talk a bit about the central dogma theorem. So this is kind of the heart of biology, of molecular biology. So as probably most of you know, or DNA basically encodes the building plan in all living organisms. And the central dogma theorem basically states, well, there is DNA, this DNA gets transformed into RNA, and this RNA then gets transformed into proteins. And these proteins basically are the base of all our living things. So everything in your body is basically more or less built out of proteins. And this is why this is such an important topic. Yeah. Just a quick recap. So what is the DNA? So basically you have this, basically normally one reads this from this 5' end to the 3' end. And it's basically a code expressed by these bases, nucleotic bases they're called. So basically you have these four here. You have adenine, guanine, cytosine, and thymine. And basically the way it works is that only one can only have an opposite partner, which is always fixed. And so you can read this in both sides. And basically you can open this and replicate it, because you get always the right partner here. Yeah, but this is just one part of it. So there is the DNA replication process. The structure of all of this has been discovered in the 1950s, and basically this is where molecular biology starts. So in the 1950s, basically we start to do all these kinds of sciences. Yeah. So we have this DNA, and basically then it goes over into RNA. So RNA is basically the same. It's just the adenine. So basically the thymine here is replaced by a different thing, which is called the uracil. So you have one different partner, and then from these, basically you have this genetic code, and with this genetic code, basically you can build proteins. Yeah. Why am I all telling you this? I don't want to bore you with this, but I just want to get everybody on the same level, everybody on the same side, so that you understand what's really going on here and why this is also important. Yeah. So basically you have then your RNA, and as we said, we have these four nucleic bases basically. And for instance, let's say you have a uracil, adenine, and adenine, and then you have like this stop codon. So basically this means here you stop reading, and otherwise you build amino acids. Yeah. So basically if I have UCC, then I have this S amino acid, serine, I guess it's called. And basically you read this, and as you read this, basically you build a string. So this folds into proteins. Yeah. So how this works, basically you have this peptide bond structure. So you have, this is always like one of these amino acids, so on the outer side of the code. and they form together a chain. Yeah? So as I said, you read basically the genetic code 5' to 3', 3', and then basically you get into these amino acids. And these amino acids, they form these bonds, and basically it's just a long chain with different residuals here, and these residuals, you have to form them in order to get the function. Yeah. So what does this look like? So these is what these residuals look like. And what we basically want is we want to know the structure of the final molecule, because only if we know the structure of the final molecule, we basically can see what it's useful for. Yeah. And in the old days, basically what you did is you watch this table that I show you here, and from this table you get some properties, and from these properties you somehow know or have some kind of intuition what's going to happen, because well, if the side chain, for instance, is hydrophobic, then you can say, okay, the way this has to fold is that the hydrophobic parts, of course, they don't touch the water, so they should be on the inside of the molecule and not on the outside surface of the molecule. Yeah. And you could, for instance, say that maybe positive and negatively charged ones should be facing each other and things like this. And this way, in the first years, so let's say from the 1950s onwards, people started to fold this by having these kind of ideas of what things look like, and this worked more or less well or not. Yeah. So this is the story about protein folding, and as I said, so basically, when you fold them, you kind of get the idea of what the function works like. And also what you should know is that these proteins, they're not static, but they're also flexible. Yeah. So even if you have a fold, or if you think you know the fold, it can always happen that the protein during its lifetime changes its folds and goes into different states. Yeah. So this is the whole problem that basically happened in this molecular biology. And just to get you an idea, so here you have like the complete picture of everything. So we have basically these flagellars. So let's see it like this. We have like a bacteria here. And this bacteria basically has these flagellar strings here, which it uses in order to move. Yeah. And as you can see, so the way it on a molecular basis looks like, so here you have this like on an atom scale resolution here. You have these all kinds of proteins which build kind of the motor here in the cell membrane. Yeah? So it really looks like this. And this whole thing here can turn. And when it turns, basically, it induces a movement into this flagellar. And this way, the E. coli bacteria basically can move around. Yeah. So just in order to make this work, we had this genetic code. So somewhere it's encoded in the DNA. And then we just basically, we don't write the inner code, but if you have proteins, you can write the outer code, which is basically equivalent to the inner code. Yeah. So basically, this is the string that we have here. So this is the sequence. The sequence, this sequence here, it folds into this structure here. This is on the atomic scale. So basically here you have the real, so basically each line here is a bond and connecting atoms. And here you have this in what is called the secondary structure. So this is basically just an image for biologists, or so that they know, because there are kind of recurrent structures in these proteins, how basically hydrogen bonds link these amino acids together and form these recurrent structures. So you have these coils and these beta sheets here, but this is basically the same. And then just to give you a perspective, so this little piece here is the green piece that you have down here. And you see you have this like a multitude of times, and then you have a lot of other pieces, and you have this whole machinery here. And so the basic idea is, well, how from DNA you get to this kind of machinery. So how do you know from what's encoded in the DNA that this part of the DNA builds this machinery, and what does this look like? Yeah. So this was a huge problem basically to be solved. Yeah, and this is where AI basically revolutioned the whole story here. Yeah. Just a quick check. Do you still hear me? Everybody hears me here? Yeah. No feedback. I'm a bit worried. Okay. You're still here, everyone?
Yes, Professor, all going
Okay.
well.
Perfect. 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 So so this is the basic story. Yeah. Any questions so far from your side, from the students, to this whole story? Okay, then I just continue. So basically, we have this
Yeah,
sequence.
sorry for the interruption. So if
Yeah.
anyone has any questions, feel free to add them to the chat and we'll read them out loud to the professor.
Okay.
Okay? Just to make it clear
Okay.
to everyone. Thank you, Professor. You may proceed.
Okay. So basically, we have these sequence to structure. It's of course a bit more complicated than just translation. And as you see, basically, it's very complicated to get from this string to know that this string it folds into something like this, and then this place basically builds a part in this whole machinery here. Yeah. So it's even more complicated because there's gene regulation going on, which is called in these terms omics. So gene regulation basically means that, as you know, basically in all your cells, you have the same DNA, but of course a neuron does something different, as I write here, than like a cell in the liver. And how does this work? Basically, you have something which is called like cell differentiation. And in each cell, basically different parts of the DNA are read. Yeah. And this is all most of the time in part steered by epigenetics and by this whole omic story. So basically there's like a gene expression profile, it's called. So it depends on which parts basically that you read of the DNA, what the cell actually does. Yeah. So the cells can be in different types of states, and even during their lifetime, the state can change. So we will go over basically the white blood cell, which can, in its lifetime, do all kinds of different things and change its DNA expression profile because of what it does. Yeah. And all this, you also have to understand it if you want to go into this protein folding things, into this kind of stories. So about protein synthesis, I will keep this movie for you. You can watch this later after. Maybe we can share the slides with the audience. This is a very nice talk, basically, which from 1971, or basically a show where they very nicely explain how proteins basically are synthesized with the whole molecular machinery, from how you get basically from DNA to protein. Yeah. Okay. So far so good. Then we also have a data integration problem where AI basically can help us. So the story is that the number of sequences is dramatically increasing because it's becoming so cheap basically to sequence gene sequences. So just in comparison, so when the first human genome was sequenced, so this was done between 1990 and 2003, in the Human Genome Project, it cost estimated like $3 billion US. Today, if you want to sequence your own DNA, you can do it for around $1,000. You can order, basically, you spit in some tube and you send it to some company and they send you back your DNA to download on your computer. Yeah. And this way, of course, we have way more sequences from all kinds of animals, from all kinds of bacteria, and we have somehow built a machinery, and basically we have to treat all this data. Yeah. For instance, if today you write an algorithm which tries to infer evolution in all these sequences, well, it has to be way more performant because the number of sequences, basically, it grows faster than our computational power. Yeah. So we have to build always more and more performant algorithms in order to treat them. So how did we do things classically? Yeah. What did people do before AI and this AI revolution came, how did we resolve protein structures? So you already see what we are going to. So basically, we will see that we can do this protein structure inference with AI, basically. But in the old days, basically, we did X-ray diffraction. So you use these kind of proteins and you let them crystallize. So crystallize means basically you put them in some kind of water, you let the water evaporate, and with good luck, with some chemicals inside, crystals form. And then using these crystals, you can do something which is called X-ray diffraction. And you can read these diffraction patterns. And from these diffraction patterns, basically, you can get the atomic coordinates out of the molecule. This is how the first DNA was basically found, how we found out that the DNA is this double helix structure in 1953. Yeah. You can also do this with NMR spectroscopy. So NMR spectroscopy is basically a resonance system. So you send some impulses on the molecule, and the molecule basically responds to these impulses. And from these impulses, you can also infer the structure of the molecule. And then you can do electron microscopy, but electron microscopy is like a half-baked thing because the resolution is not good enough, so you don't really get on an atomistic level, so you don't really know where the atoms exactly are, but you kind of get the molecular envelope. So you know basically, okay, from the outside, the whole molecule, it looks like this. And then we did, of course, also before AI, we did some molecular dynamics and in silico folding, so in the computer folding. So basically, you took this string of amino acids and you kind of ran a computer simulation in the hope that it folds into the protein that you wanted to have by putting physics equations behind it. Yeah. What do we need this all for? So all of this is very important for drug design. So here a quick example for that everybody knows, but this is basically the receptor for adenosine. So adenosine is like a byproduct when you use your muscles. Basically, you produce you burn basically ATP, so which is a molecule which gives the body energy, and this produces this adenosine. And if you have a certain level of adenosine, it's captured by this kind of molecule here, and it triggers that you get sleepy. Yeah. And if you look at the structure here that you have in here, it looks very similar to the caffeine molecule. And what can happen basically is that if you drink coffee, that the caffeine molecule gets bound in here, and the adenosine basically can't enter, and therefore this receptor doesn't work. Yeah, which keeps you basically awake even if you should be tired. Yeah. And this way, if you understand these kind of structures, you can, of course, not only for this part, but for all kinds of mechanisms, you can steer them and build basically molecules or basically drugs which could help you to, to overcome diseases and to do all kinds of things. Yeah. That's basically the story. So right now we go to already to, so these are the basics that I wanted to have so that everybody is like on the same table, that you kind of know why this is also important. And right now we come to AlphaFold 2 and AlphaFold 3. So basically AlphaFold 2 was the big breakthrough in this protein folding story. So there is this Levinthal paradox. So before that, we said, okay, people kind of gave up on it because they said we will never achieve it, that kind of computationally we can really fold proteins. So the idea was basically this guy, he said, well, if you only have 2 amino acids and there would be only 2 folding variants, so you can basically show that if you would then extrapolate this to 150 amino acids, you would have 2 to 150 folding variants. And if you want to check them all, even if it only takes you, I don't know, a femtosecond, it would take you like 10 to the 24 years in order to find the fold. Yeah. So this is kind of why people gave up on it, but AI kind of came to help. Yeah? So the people never really gave up, but there is like something called this critical assessment of techniques, basically how to find protein structures, so from the DNA sequence. In the 40th conference, basically this AlphaFold 2 came up, and what they do basically is in this conference is they have put out known structures that were recently discovered without revealing them. So they sent out sequences to the participants. They know the structure in their lab, but they estimate that the public does not know the structure. Yeah, and then they ask the participants with all their computational algorithms to predict what it looks like. Yeah. And the idea is that, of course, then you can score how well these predictions were made. And AlphaFold 2, so basically the AI, it arrived at 90% accuracy, even more than 90% accuracy at these structures to predict them. Yeah. And this is basically indistinguishable with experimental results. So what the experimental results is basically what I've shown you, X-ray diffraction and what we did before. And this way, basically, the problem was solved. Yeah? So just to give you a perspective on what a breakthrough this is for science. So in 1958, so I told you in 1953, DNA was resolved. So it's kind of the same problematic a bit. But so a few years later, because as I told you, you have this DNA. From this DNA, you get the RNA. And from the RNA, you get basically the protein. And this little protein here, with this little antagonist here, was the first protein to be resolved, and basically by John Kendra and his team in 1958. And they did this also by X-ray diffraction, so by the same method, basically, that the DNA molecule was resolved to be a double helix. And from 1958 to 2020, by the same kind of methods that I've shown you before, around 150,000 structures have been resolved, okay? And no further. So basically this 150,000 structures, they have been used to train AlphaFold. And AlphaFold was so good, basically, in predicting new structures from the sequence that it compares to these, basically, to these traditional methods. And it allowed us since 2020, so in the just last 5 years, to have more than 200 million structures that have been resolved since then. Yeah. So just in perspective, so basically it took us 70 years to get 150,000 structures from the starting that we had the idea on how things work. And with AI, basically, we got from these 150,000 structures to 200 million structures that we could resolve, and that people can right now use for protein docking, for in order to understand how drugs works, how our body interior basically works. Yeah? Just to put this into perspective. So, of course, we're academics and we want to know a bit how this whole thing works. So what AlphaFold does and how this works. So basically they use a transformer inspired architecture. So the transformer, I guess all the people who are in AI or who heard the talk about AI, basically they know what it is. So it is this kind of attention mechanism that LLMs use, so that you basically know on the current word, basically also which on what kind of words you should take into account in a phrase. So it kind of gives you an idea about what's happening around you. It basically, it uses this attention-based mechanism, and it uses it twofold. So first of all, it does a sequence alignment. Yeah. So basically, when you put a sequence in, it has huge databases of sequences that have been derived, that have been sequenced, as I've shown you before, and it aligns them to all of this. Yeah, and this allows it basically to work with the evolution of the sequence. So the idea is basically that for the cellular mechanisms that you have in your cells, most of them are conserved. Yeah, so for instance. I do DNA replication in my cells the same way as a cat does DNA replication, and even almost the same way as a bacteria does DNA replication. Yeah, and there are kind of the same molecules involved, and these molecules, they undergo evolution, but they still stay somehow the same in their fold. Yeah. And if so, basically one amino acid changes in the evolution, some other amino acid also has to change in the evolution so that the fold kind of stays the same. And this kind of goes into this alpha fold mechanism in order to understand better what the structure, the end structure will look like. Yeah. And the second thing is it takes like triangular distances. We will see a bit how this works. Of geometry, so initialized by geometric homologs and then refined in this transformer-inspired network architecture in order to basically build the protein structure from the sequence. Yeah. So this is this kind of the story here. So as I told you, why aligned sequences? Basically, it gathers the evolution in this whole system. So it basically it knows, okay, there are other structures that have a similar sequence, but they look a bit different. So I can infer, okay, if this amino acid changes pairwise with the other amino acid, that even if these two change, I have to retain the same structure. This is kind of the story, and it also gives you an idea. But if this one changes at the same time as the other changes, that these are probably close to each other. For instance, if they're charged. Yeah. So this is kind of what goes into this network. And then we have, of course, this triangular mechanism. Yeah. So as I said, there's also this structure information with these triangular systems to take this into account. Yeah. So both parts basically exchange information. So you have this structural part and you have this evolutionary part. And from the evolutionary part, of course, as I told you, you get information to the structural part, but also from the structural part, it kind of gives back according to the triangle inequality. So as I told you, it takes these triangular systems into account. So if basically the triangle inequality doesn't work, look, even if you have a good idea here in the evolutionary system, just stop it. In the structure, it does not work. Yeah. And this way, basically, you get the structure at the end out. So to give you a quick overview, this is kind of taken from the paper, what it looks like. So as I said, you have your input sequence, and the idea is that you get the fold out at the end. Yeah. And this is the big breakthrough here. So you type here your DNA sequence, and on the other side, we have basically the fold. Just give me a quick signal. Does everybody still hear me? I'm always a bit worried because sometimes the microphone kind of get lost in this system here.
Yes, Professor, we can hear you.
Okay, perfect. So basically we have the input sequence, and then we make this genetic database search. And basically it gets all the close sequences to these, as I said, and then kind of it knows, okay, if this one here has changed and this one here has changed at the same time, these two have to be kind of close to each other. Yeah. This is, which of course gives them an implication into the structure. Yeah. And on the other side, you have basically a structured database search, and you have here like a distance matrix where it says, okay, this one should be at this distance to this one, and this one should be at this distance to this one. Yeah. And this kind of network here, in there, they also use these kind of triangular approaches that I talked before in order to get you back to the structure. Yeah. So here a bit more a close-up view. So as we said, we have this kind of sequence alignments here. They feed basically back to the structure, and the structure basically feeds back to this at the entry here to this sequence alignment part. And here you have this kind of triangle update. So the way this transformer network works is basically that it always sees this matrix here with these triangles. So here you have one amino acid, here you have another amino acid, and here you have a third amino acid. And then it kind of updates them together with each other, with taking basically attention into account between these trees and between basically blocks of these trees. What's important here is maybe to understand this better. These triangles basically they go over almost all the atoms in the system, but it's not like that. It's like connected in a chain. Yeah? So they basically in this structure they ruled out basically this chain connection because it performed better if they didn't have this chain connection, but if they just had these triangular connections basically going on. Yeah. And they do this 48 times, and then they have some kind of machinery back here, which basically from these triangles and from this MSA representation then builds the final structure. Yeah. That's basically how it works. They trained this, and basically they won with this ESP 40 story. Yeah. So this is of course a huge breakthrough, as I told you, but it also has pitfalls, so nothing's perfect. So there are still, of course, sequences where you might not find a lot of homologs in these genetic databases. And there might also be sequences where you don't find basically a good structural information. So this AlphaFold 2 algorithm, it's very much important to have this kind of genetic databases here. This one is not so important, which is kind of intuitive, because otherwise, I mean, you want to predict the structure. You don't do homology modeling, what we already did before, where you basically say, okay, well, the sequence, it looks like the sequence of another protein, which is very close to it. So it must have kind of the same structure, but of course it helps. Yeah. So what is really important is this part up here. But sometimes it can happen, you want to build a new sequence, or you want to do something very different, that you don't find basically similar sequences, or in the evolutionary history something that is really similar, and then this kind of can fail. This is one thing. And the second big problem, even more bigger problem, let's say it like this, well, in general, this system here, if you don't tweak things, I mean, with some tweaking, you get maybe one or two structures more out of one sequence. But the problem is that in general, you can say you can get one structure out of this. Yeah. But as I told you, well, in real life, these things are flexible and they can refold or they can move. So basically, it can happen that this coil here goes up here, or basically does something different. Yeah. So you get one structure which is very good, but it doesn't mean that you kind of get all the possible structures that you can get from this sequence. Yeah. So this is basically where the story basically fails, yeah? And where you still have things to do. And this is kind of where we then come to the next part. So why molecular dynamics, for instance, is still important, because we got one fold, but we want to see basically from this fold in what kind of other fold can we form this molecule and things like this. Yeah. How can you make use of this? Well, the model is open source, so basically you can download this. You can run this on your machine if you find a machine in the lab. It looks a bit large, but it's not like I mean, it's a machine that a lab should be able to afford. So you need a GPU, only one GPU, which is not much today, and you need a rather big SSD. They ask you to have a SSD because you have to look up all these homologs, and I guess basically the genetic search database, it's like 7 terabytes from what I read, something like this, and they want you basically to have at least 4 terabytes of this on an SSD. And you should have like 64 gigabytes of memory. But if you have this, as I said, this is a machine that you can buy. You can basically run this whole system machinery on your own computer and make your own structure predictions from a DNA sequence. Yeah. An easier way to use this is basically there is the database. So as I said, we have like these 200 million predicted proteins right now from all the kind of sequences that are out there. So if you work with a human protein, as the human DNA is known, probably it's in this database. If it's not a very flexible protein, where it basically can't make a prediction. And you can just look basically the predicted structure up in this database and use it for your research, and then run molecular dynamics or anything else behind it. This is kind of the story of this. So as outlined, basically it's not perfect. So for flexible systems, there are like also unstructured proteins. So there are proteins where just basically this chain, it swims kind of around in the cell and does all kinds of things, where basically this whole system does not work. Yeah. And there are regions that are just not predictable, or because they're very flexible, and there are no similar things, because flexible systems, of course, they never crystallize, they never were kind of in a training set or something similar. So and of course, as they're flexible, they can fold into almost anything. So it's hard to make a prediction for them. And what they do basically is they give you a score for each amino acid on the structure, how well it can be predicted. And you can just use this score to know if your system kind of makes sense or not. So what we have here, we have this confidence score. So there is a local confidence score. The local confidence score basically tells you, well, is the amino acid here in the right position locally? And then there's also kind of global confidence, which tells you, well, does the whole fold of the protein make sense or not? Yeah. This is the important thing here to know. It can be that the local confidence scores are very high, but the global confidence scores are low. This is also fine, because then you can say, okay, there are some domains on my protein that are very well resolved, but there's maybe some linker region, and as this linker region is flexible, well, basically one part of the protein could turn around the other part or something like this. So you can basically look in these two scores and make out of it something in order to say, well, is my protein correct for what I want to use it or not? Yeah. That's what you should take into account. So as I said, AlphaFold 2 was basically a huge breakthrough. The group, so it's basically the group who did this, it's called DeepMind. It's a part of Google, who built this whole system. There's also, they iterated on this and they built something called AlphaFold 3. And AlphaFold 3 is also very interesting still, because what AlphaFold 2 was not really built for, it was built in order to get these structures. But as I said, well, the proteins, they can deform. And if one protein basically finds another protein and docks on it, in order for this docking to happen, maybe it deforms already. Yeah. So it's way better, this AlphaFold 3, at predicting like protein-protein complexes, protein ligand complexes. So protein ligand complexes are like what I've shown you before with the caffeine molecule, as well, typical protein ligand story. So basically, if you are in drug design, you can take your drug, you can take the protein that you believe it interacts with, you can put both into this AlphaFold 3 machinery, and you get like a structure where the ligand is probably in its right binding site, yeah, which is tremendous for people who do drug design. Yeah. You can do protein DNA interactions. Yeah. So there are proteins that dock to DNA. We will also talk about this when we go into epigenetics. There are protein RNA complexes. RNA themselves, it folds into structures and can basically also have kind of protein-like functionality just by folding into structure. Yeah. These are basically the things that we have, and it's less dependent on sequence alignment. So it works on a different structure. It still takes the sequence alignments into account, but it's less dependent on them, which is an advantage. The huge disadvantage of this whole story is, well, a contrary to AlphaFold 2, which you can more or less just run on your machine right now. It's not open source, and it has kind of a limited usage to academics. Yeah, so basically Google takes this AlphaFold 3 as their own property, and they want to use it for themselves in order to sell it to drug companies. And therefore, we don't have kind of the same usage power that we have with AlphaFold 2, where basically we can just use it the way it is. With AlphaFold 3, you more or less need a contract with Google in order to use it. Yeah? You can use it through a server, but then you have like limited throughput. And if you can like predict, I don't know, one structure a day, that's not what you want to do. If you really are in this field, you want to use it like maybe you want to predict the hundreds of structures a day. Yeah. Especially if you do like, I don't know, protein ligand complexes or things like this. Yeah. The key differences between AlphaFold 2 and AlphaFold 3, just to sum them a bit up. In the AlphaFold 2, they call this the evoformer structure. Here they use a different architecture, which is called the pairformer. So pairformer, basically they adapted the structure that you can have these protein ligands, protein DNA, protein RNA complexes. Therefore, a pairformer structure in order to take basically two sequences or a sequence and a molecule into account in order to predict the structure. And it uses not a poor structural process like we had in AlphaFold 2, but it uses a diffusion-based process. So a diffusion-based process, we know it basically, it's kind of the same process that is used in these generative AI models, in this artistic models, so where they make like this image generation and video generation that you know. So basically, they start out of some kind of random values, and then refine these random values step by step into the protein structure. And with this diffusion-based process, basically they were getting better not to use not as much these these aligned sequences that I talked about. Yeah. It still is not perfect, so it still lacks this conformational flexibility. So conformational flexibility means getting a lot of protein structures out for one sequence. We still don't have this. It's also very bad at discovering multiple states of a protein or the right state of the protein. So this is rather problematic because, for instance, if you have like a protein ligand complex, then in several times, basically the protein has to be in some kind of special conformation in order to accept the ligand. Yeah. And it might put, for instance, the ligand on the right side, but the protein still is folded in a different state. So you get a bogus output, basically, that doesn't really confirm what really happens in reality. Yeah. These are basically the problems that we still have with AlphaFold 3. Yeah. That's basically it for the for this kind of structure prediction part. Are people still here? Do you want to give me some questions before we go to the molecular dynamics part? We have like 3 parts. These parts are simpler and less maybe shorter than the other ones. We kind of like have like, let's say, half of the talk through from my way or something like this. Do you want to pose me questions? Do you still hear me? People, you're still there? A bit worried.
Oh, yes, Professor.
Yeah. Okay.
Yes, Professor, we can hear you.
Okay.
Yeah.
You want to push me out some questions if there are some, or I should just continue to the end?
So no questions so far in the chat.
Okay.
However, if you want, you can continue to the end, and then if anyone
Okay.
asks anything,
Okay.
we'll
Okay. So then
ask
I will
directly.
continue. So we have this molecular dynamics story, basically, which overcomes kind of, or is here still to overcome this kind of flexibility issues that we have, and this discovering multiple states of a protein. And basically this works. I don't know. I know that the host, basically, he was at the course that I tried to do, but then the war kind of broke out on molecular dynamics. So you know a little bit about it, but basically also here AI can help. So molecular dynamics, what is it? Basically, you have people who come from physics, they know all this. So you have kind of a potential. And you kind of model this potential, and with this potential kind of you can integrate it and derive basically molecular movements out of them. Yeah. So just to tell you, basically you have here, you can just take these two atoms and put inside like a spring potential. So basically with a spring constant, which keeps it at the same radius. And if right now a water molecule basically bumps into it from the side, basically it will go in there and then bounce back. Yeah. And this is actually what happens in reality, but of course not with a spring potential. So the spring potential is kind of a replacement of the actual potential. And you can do this for the angles, and you can do this also for these torsion angles. And then you can use these kind of non-bonded interactions, of course. So this is like the Coulomb interaction. So if you have two charged molecules, you have this Coulomb interaction. And you have here these van der Waals terms, basically, which are also non-bonded terms, which introduce forces between the atoms of the molecules. Yeah. And if you have basically this system up here, you can just take the gradient. From the gradient, you get forces out of it, and you can just basically, as you know, these are the accelerations, then kind of integrate all this up and get the dynamic system on how the protein kind of evolves in time and refolds in an aqueous solution, yeah, for instance. Yeah. I brought some movies with me. I hope it works through the network, that you will see them and that you don't have like a slideshow if I play them to you, just to give you a little insight on what this looks like. So here, basically, a drug design story in molecular dynamics. So here you have kind of a membrane thing going on. So these yellow particles here are the phosphorus atoms of a molecular membrane. So these are like fat acids here, which build a molecular membrane. And this is kind of a transporter protein, which transports things out of the membrane into the exterior of the membrane, so that these, let's say, toxins can't get into the cell. And basically you see how everything moves, how everything wiggles. And I mean, you see the molecule, yes, these two parts down here, they kind of change their conformation, but they don't change so much. What was basically here to prove was that this molecule here can enter here. And so we kind of pulled it in, and you can see that it basically works. And we have another movie where basically it doesn't work, that this molecule here moves in. And so we can basically say, okay, this drug might work in order to block this system, and the other drug might not work. Yeah. That's the story here. This is kind of what you can do with these molecular dynamic stories. And of course, if you have AlphaFold, it's very great, because you know already this structure here, the starting structure for this molecular dynamic simulation, and it gets you basically quicker into working. Yeah. So far so good. Where's my presentation? Let's go back to full screen here. And basically, but we're not only talking about molecular dynamics, but about molecular dynamics and AI. And there are several things that AI kind of tries to improve in this molecular dynamics. So the integration between the algorithms kind of hard, because these molecular dynamics or codes are already a bit old, and they're mostly written in C and C++. Also for performance reasons, and even they run sometimes on custom hardware, so they don't run on GPUs like what we do with AI, but they even have custom hardware built for them, and to bolt on an AI system on these systems is a bit complicated. Yeah. And there's an effort basically to implement these MD algorithms in AI frameworks. We will go into this, how this should work. Then we have kind of AI-derived force fields, so what I've shown you before, and we have, of course, AI and machine learning in trajectory analysis. Yeah? So these are kind of the three parts where AI kind of has a field. So I took this picture here from this article here, where they show how they want to try basically to implement molecular dynamics in an AI manner. So what they do here is they make this kind of comparison, which they're somehow right in what happens, because as you saw, you have to take this kind of gradient here of the potential in order to get these forces out, which you integrate, and then basically you get a new coordinate system, and then you kind of repeat this process. And basically what they say, look, well, if you look at it, it kind of looks similar to the way how you train an AI model, because in an AI model, basically you have this loss function. So the loss function is kind of how good you are, and then you do this kind of back propagation, and this back propagation is basically you calculate the gradient, how you should adapt the weights in your model in order to get better outputs. And so you have all times these gradient calculations that you have kind of here too. And they say, look, this circle basically is kind of the same. So we can use these AI machinery. So the same machinery that we use in AI training in order to implement MD simulations. Yeah. And they do this basically. So there is something which is called torch. I guess I wrote it here. Which is called Torch MD. So where basically in Torch, they implemented with the Torch machinery an MD simulation code, yeah? Which has the advantage that it's not anymore in C, but in this Python land, what the Torch kind of lives in. And of course that it runs on GPUs and probably faster on GPUs than classical MD simulation codes. Yeah, but it has all kinds of downsides because these MD simulation codes are very old, and there's a lot of knowledge inside that kind of didn't make it into this algorithm, of course. Yeah, but as I said, this is a landscape of active development right now. Yeah. Then there are neural force fields. So basically what people do is they don't take kind of this V here, but basically they take snapshots of existing simulations that you can do on a higher level. So you can kind of, instead of doing all this, you can use kind of quantum mechanics and say, okay, I do this in quantum mechanics and I get forces. And for these forces, I built a neural network which kind of gives me so that I can kind of learn this potential here. Yeah. So they learn this potential out of a landscape of forces. Yeah. And they do this with a neural network. So, and this is called neural force field. So this is something that you can do. As I said, this allows you to use higher level dynamics basically on a faster and make the system a bit faster. Something very important, maybe not very intuitive, but I will try to explain you this way. Basically, when you saw the videos, you saw there are lots of things going on and they are very fast. So you have very fast movements, and all these very fast movements, basically these Brownian motions that you have in your MD, they're not really important. So what you want is kind of these slow motions that happen in the MD simulation, like this rearrangement of these two parts of the molecule that are like on the downside that I've shown here. Yeah. And in order to get these slow collective motions, basically you want to make like a coordinate transform, which kind of gets you rid of all these fast movements and only shows you these slow movements. So if you walk along this coordinate, that basically you get this slow movement of the protein. Yeah. And in order to find these collective variables, so this is called this set of new, let's say, of new variables that kind of it's a dimensionality reduction process. Yeah. So if you know about PCA or a principal component analysis or algorithms like this is kind of the same idea, but here you just do it with AI basically, or with deep learning algorithms. Yeah. And you do it so that you basically get these, as I said, that you get these kind of slow movements and filter these fast movements out of the simulation. Yeah. So as I said, in the old days, we did this with PCA in order to find variance maximized variables. So basically, which then tells you, okay, well, if a lot of atoms move at the same time, then it's kind of probably an important movement. Yeah. Today, you can do this with so-called time-lagged autoencoders, and they hopefully do this better. So how does this look like? So you basically you take your simulation at time step t, you put all the atom coordinates at time step t inside. You have this time lag, so you put the time t a lag time later. Basically, you put all your coordinates, and you build this autoencoder where you basically break to a lower dimensional system. So if here you have like millions of atoms, and here you also have millions of atoms, maybe here you only have like 5 values. And from these 5 values, you rebuild what happens kind of at time t in the future. And hopefully, basically, it filters you out these slow motions because you're kind of at the time scale of this tau and not on one frame to the next time scales. And this way, kind of, you can discover these latent space coordinates. So basically a coordinate system with a few values, which kind of gives you where the important motions are in your system. Yeah. This is basically what we do with what you can do with AI on molecular dynamics. Yeah. Something else that you can do that I've developed, so I want to show it off basically. So basically what we have here, this is kind of machine learning, but can also fall into the AI field. So when you do this molecular dynamics, basically you have your potential landscape, and in this potential landscape you have potential wells. So here in this schematic two-dimensional part here, and in order, basically, when you run your simulation, most of the conformations get somewhere in these wells, and then it takes you as kind of energy to hop from one well to the other, basically to induce a conformational change. And you can kind of sample this and use a density-based algorithm to find basically these dense clusters. And if you relax the density, basically these clusters here get formed together, and you kind of know, okay, how far is the 1 to the 2 and how far are they basically to the 3, and have this in kind of a tree-based structure. So you see here intuitively from this diagram that 1 and 2 are kind of closer to each other, and that if you induce more energy into the system, you can also get to the 3 state. So then at a different stage, you have kind of the 1 and 2 and 3 state. Yeah. And basically, you do this with the TP scan algorithm. I will just look at how much time we have. I guess we already are a bit out of time, so I will very quickly go over this. I will not really tell you how it works. It's not important, but what you basically get out is kind of this tree where you have all these kind of different states, and where you basically know, okay, you see the simulational movie, but you know basically, okay, well, here I'm in this fold state. I need this kind of energy to get from the B to the C state. And if I want to get from the C state to the, I don't know, E state, I need that much more energy to get over to the E state. This is something that you can do, yeah. This is one story. The other final story that I want to go into, even if we don't have so much time, is I want to talk quickly about LLM technologies in sequence prediction. So, of course, what is also important is like what happens basically if one of these sequences, of these gene sequences, undergoes mutation, especially if you kind of are into hereditary diseases, for instance. And kind of how can I understand which mutations are important or not? And in order to do this, you can use these kind of sequencing technologies. So basically, you kind of use the same machinery as you have in an LLM, but instead of predicting word pieces, you just predict basically the next base in the next DNA code, if you want. Yeah. And what you can do basically with this is you can kind of when you have this kind of machinery, you can put in a gene sequence, and it then tells you, okay, what is the probability of the next part of the sequence. Okay, and if for instance, so let's say the next letter in your genetic code should be A, yeah, um, then you have a certain probability to get A. But if basically you get C and the probability to get C is very low, then you know, okay, basically probably as the probability for C is very low, as this is trained on a lot of genes, and also what I told you before, an AlphaFold goes in here, um, then probably this mutation will break the protein's function. Yeah, this is the idea behind it, because you can basically say, okay, you have a certain probability to get basically this kind of base or amino acid here in the sequence, but you get another amino acid. So if the probability does not match, well, then probably your system will fail. But if the probability still matches, then basically the let's say it like this, then the change is okay and it doesn't destroy the function of the protein. Yeah. This is something that we have. We also still have epigenetics, but I guess as we're already at 10 after 6, I will stop here and not go into this epigenetics part. Epigenetics would also be interesting, but maybe we can do this another time, or it's fine here. I guess but the most important thing that I wanted to show you basically was the whole AlphaFold story. As it's a real scientific breakthrough. The people who built this, they got the Nobel Prize for it. It's done with AI, so AI is not only ChatGPT and these kind of, let's say, tools that we use every day. It really brings breakthrough to science also, and that's where I basically will stop here and answer your questions if you have some. Yeah.
Thank you,
Are we?
Professor, for this wonderful talk. It
Yeah.
was really, really insightful. Many thanks again for joining in this session. So, if anyone has any questions, please feel free to share them in the chat. If not, I will start it off with something relatively simple and straightforward. So, let's assume that everyone that's attending this is a computer science background or from an AI background per se. What would you recommend that they should do if they want to dive into the world of bioinformatics, considering there's a lot of details to be known on the biological essence of the problems that they're trying to solve?
Uh, I guess it's not so hard to get into it. Yeah. I mean, it sounds like there's like a entry curve, but I guess the entry is way lower than, let's say, in biology or in these biological systems than getting into physics. I mean, the story is like what I find interesting about biology is like an unsolved problem is like around the corner. So you very quickly go into things that are kind of unknown. While in other disciplines, probably it takes you a very long time in order to get to the part where kind of, okay, right now I'm at the frontier of science. Okay, in biology, you step in, you learn a bit, and maybe after 1 week you go to frontier science. Let's say it like this. Yeah, it's really like that. It's I wouldn't have people fear that the biological part is so complicated. If you're in computer science and you want to understand this, I think the algorithms are all the same. Yeah, it's like this. Yeah, it's like what's important is that you know the algorithms, that you kind of have an idea biologically into which direction am I going, that you understand the central dogma. Okay, that's fine. That you have kind of a bit of an idea what's going on, but it's not that let's say not that hard. Let's say it's hard, but it's not that hard to go to real science. Let's say it like this. Yeah, I mean, I can just say it from the experience that I had. If you're in physics, for instance, okay, it takes you years of study And after years of study, maybe at your postdoc, you come like to a problem where you say, okay, right now I'm at the frontier of science. Right now I'm writing new physical laws that have not been discovered before, or rewrite the ones that have been discovered. Okay. In biology, it's like you start and you're right at the frontier because there are so many questions that are just unsolved. Yeah. Does this kind of answer to the question?
Yes, Professor, it makes sense. Something along the lines of what you explained is, so can we so you mentioned that there are a lot of interesting problems, or not, maybe not problems, but downsides or things that are still not solved when it comes to AlphaFold, for instance, AlphaFold
Hmm.
3. And would you say that this is the main biology problem that's currently trying to be solved, or would you say that the attention should be, or currently is directed towards another major problem, similar to how protein folding was the major thing that they were all focused on? Is there another direction or another problem on the horizon that everyone is directing their attention to?
Yes, so there are several, as I said, I mean, there are different things in biology going on. And I guess there's like in computational science, in computational biology, there's like these two different groups. So there are like the people who run on structures, so they're into AlphaFold, these kinds of problems, into molecular dynamics. And then there are all the sequence people. And I just talked about the sequence itself. So I started a bit basically that today there's a lot of things going on. So if you're interested in this other part, there is something called the ARC Institute. So it's written ARC Institute. And this ARC Institute is probably right now on the frontier of science on the sequence part, where basically they built this model that I've talked about, where you can like from a short part of a sequence can predict how the sequence continues, and this way you can basically find out if there can be mutations and if these mutations are important or not, if they will break the protein or not. This is kind of one part, and this whole kind of epigenetics, omics parts. Yeah? So epigenetics, omics is like what I told you. If you have a cell, there are these whole all regulation stories. So is a gene expressed? Is a gene not expressed? If one gene is expressed, what does this mean for the other genes? Yeah. So basically, these people, they are more into network science, and they basically look how all these things regulate each other. Yeah. Because it's like, let's say it like this, and a cell, it has kind of limited type of energy, and if you express one protein, you don't have energy to express another protein, for instance. Yeah? And by this way, there's a whole regulation process going on, and if this regulation process doesn't work, this is in general how you build cancer or how you get autoimmune diseases, like for instance, multiple sclerosis, where this kind of regulation process doesn't work and the cells do things that they should not do. Yeah. And this is also a very interesting field in biology, where AI can do a lot of things, where we could have another talk about it, but I guess we're fine with what we did. And as I said, if you're into this, I will share you my slides. There are things on my slides. If you're interested, go to this ARC Institute website. I guess they are on the forefront of this kind of technology right now. Yeah. Okay. Any other questions? Okay, if there are no questions anymore, I guess, Haida, I will just send you my slides and this evening or maybe tomorrow, and you share them to the people who were inscribed here.
Yes, that would be great, Professor. Many
And
thanks
okay,
once again about for this amazing talk. Uh,
okay,
Please feel free to connect with Dr. Thomas Hashka on LinkedIn, And do you have a preference on if anyone has any questions that they want to personally reach out to you? Do you mind
Yeah, they
that? Do you
can
have,
just reach out to me. It's fine. It's fine.
yeah, do you have a
Definitely.
preference
Definitely.
of a
Okay.
platform by email?
Yeah.
Okay.
You can write me by email. You can share my email to them, or just reach out to me on LinkedIn. You will easily find me. Okay. Thank you so much. And I will share